Hello, in this video we will learn about a glutamate as a neurotransmitter. So the glutamate as a neurotransmitter and what is the function of the glutamate? Let's begin to understand. It is excitatory neuron will excite the another nerves and one half the basically and more than it releases from neuron as a signal to excite in the brain interneuron the terminal of the where the synapses occur. It is ionotropic and the glutamate is non-essential amino acid, remember about that, and our body can synthesize itself. This is the glutamate formula is the amino group and side chain are present. The structure is here. So the glutamate will synthesize from the glutamine. Let's understand a bit about the brain and where is the glutamate process and release. So this is the brain contain a primary input, for example emotion or some other things like when you are study. The caudate nucleus, globus pallidus, externus and internus, putamen nucleus and the amygdala is the basal ganglia involving the processing, and this is used for the glutamate for the stimulation. Substantia nigra, reticulata and compacta is used for this function glutamate to excite the other neuron. So the thalamus and hippocampus is here and this is the cerebellum. So the thalamus and hippocampus is a part of the limbic system. And limbic system is used for the emotion, love, hate, thirst and hunger as well as memory. Cerebrum is the primary processor for the integration and learning. So in this way, here is the hippocampus. It is look like a seahorse or cornu hominis is the king which that is related to their horn. See fimbrium and dentate gyrus which that receive the signal from the cerebrum. This is the CA3, CA2 and CA1. CA3 transmits the signal to the CA1 nerves. And this is the major example for only hippocampal neuron for long-term potentiation. This is the subiculum and the parahippocampal gyrus as well as entorhinal area are present here. This is the lateral ventricle, the temporal horn. So it is for only hippocampal neuron for long-term potentiation. Anyhow, let's begin to understand the synthesis. No, I hope you make sense. This basically is the entorhinal area will transmit the signal back into the cerebral cortex and the learning and emotion can increase, and in this way the process will increase, will lead to long-term potentiation. The presynaptic neuron example is collateral nucleus, collateral neuron 3, and the glutamine will go into the mitochondria, will produce the glutamate, and in this way the amine group will be released known as deamination and the form a glutamate from the glutamine, not glutamate, that's my mistake in the glutamate but should be glutamine, remember, and it will be stored into the V glut 1 to 3 and exocytosis will be occur, and in this the glutamate will be bound with the postsynaptic neuron, example is CA2 or 1. After binding the open sodium channel and calcium channel, the NAMDA, AMPA and kainate is so named because the agonist used to stimulate this glutamate receptor. So the sodium and calcium enter after the depolarization will occur, and long-term depolarization will lead to a long-term potentiation, the long-term memory store, and will transmit into the cerebral cortex and will give the response. Anyhow, here is the you can understand this is the EAAT channel, the excitatory amino acid transporter, which that transport back into the this glutamate into the again back or go into the glial cell, and the glutamate will convert into the glutamine So remember, the glutamate cycle is occur within the presynaptic neuron as well as the glial cells. So remember here, you can understand that is easy to understand because I will not show. The glutamate, it is excitatory neurotransmitter made up of amino acid. It is important for short-term memory and long-term memory even, long-term potentiation and neuroplasticity in the neurons. Let's learn a bit more about the channel and then we will understand about the metabotaric G receptor. So this is the channel which that is contain the receptor for glutamate. But when the glutamate will be burned, will open and the calcium will be move inside while the potassium can move outside. Will lead to depolarization. Inside will be negative, will inhibit through sodium influx to increase, will lead to inhibit and depolarization will occur and the positive charge will be occur inside intracellular matrix while the extracellular matrix is negative. So the magnesium can bind, but remember this is basically the sodium. Sodium will inhibit these bindings. So magnesium bind site will inhibit while it can be off from the NAMDA channel. So the glycine obligatory coagonist will use to promote this function. So the glutamate receptor NAMDA AMPA kinase is the name due to agonists that activate them. Let's begin to understand the metabotaric G protein. Basically, it is another way. The another cell is involving even neuron also itself. What is the function of that? Let's begin to understand and how glutamate is used for the growth of the brain and synthesis of the protein for the neuron. So the neuroprotein primary messenger will bind as a glutamate after the releasing from the glutamatergic neuron, and after bind with the G couple protein. The G couple protein contain intracellular trimeric protein alpha, beta, gamma. Alpha contain GDP is in the form of inactive. It will be activated due to ATP use or GTP use to activate after the intracellular signal from the extracellular signal. After this, adenylate cyclase will be triggered and the GTP will be activated after the binding with the alpha. After binding with the alpha, it means the GTP will be activated is will trigger the adenylate. In the presence of adenylate cyclase is the intramembranous enzyme. Adenylate cyclase is used for the conversion of the alpha GTP to the GTP into the cyclic AMP, and pyrophosphate will be released. After the pyrophosphate when released, basically GDP or ATP contained with the alpha subunit. After pyrophosphate will be released, the cyclic AMP will be formed. This cyclic AMP is the second messenger, remember. Will a cascade of phosphorylation will occur, trigger to bind with the DNA as a transcription factor such that protein can be acycled. AMPK or a MAP kinase or other type of AKT protein or mTOR trigger. On the other hand, there is a lot of protein is used for the triggering of the DNA replication, DNA transcription to produce a mRNA, messenger RNA. This messenger RNA go into the cytoplasm bag and the protein synthesis is known as the translation. The synthesis of protein will occur. That protein is basically the neuroprotein, the glial protein or other type which that repair the DNA and use for the neuroplasticity. And there is a lot of things. But on the other hand, the other cell is also involved. I hope you make sense about this. This video was about the glutamate as a neurotransmitter. So remember, the glutamate is play a role in the limbic system for neurogenesis, neuroplasticity, and the synaptogenesis. And there is a lot of things which that trigger the limbic system and the basal ganglia for inhibition of the Parkinson disease. Whenever the glutamate will decrease in brain, will lead to cause Parkinson disease. Thanks for watching.